Hi, thank you. I'm joining with a few others by Zoom. I want Ken Berns has been a wonderful colleague of mine in ILR, but before he decided to move up to ILR, he was recently the director of the Genetics Institute at the University of Florida, and previously he was dean of the College of Medicine and vice president for health affairs, which means he had the ear of the president, at least the previous president. He, among other things, he was an alumni trustee of Johns Hopkins Board of Trustees, and he currently serves as a member of the advisory board of the Institute for Basic Biomedical Sciences. And I don't know if that's at Johns Hopkins or elsewhere, but we all know Ken. Welcome, Ken. And I'm looking forward to this talk and find out more about the Neanderthal in me. Thank you. It's all yours.
Am I on?
Yes, you're on.
Okay. Thank you for that gracious introduction, John. Sorry you couldn't make it for the talk. Anyway, what I want to do today, if I can get the first slide up there, there we go, is talk to you about last year's Nobel Prize in Medicine, which was awarded to Svante Pääbo. He's a Swedish geneticist. He's about 67 years old. Has both an MD and a PhD from Uppsala University in Sweden. He was given the prize for sequencing the Neanderthal genome. That's the whole talk. The rest of it, we're going to try and fill in a few details. So he was born in 55, and his parents were both scientists. There must be something about genetics involved there. And he trained at Uppsala, as I said. Then he had postdoc studies at the University of Zurich in Switzerland, and then in the Department of Biochemistry at the University of California at Berkeley. At an early age, when he was only 32 years old, the University of Munich invited him to become a full professor of biology. And in his book, he said that it was a department where he didn't have any idea what the other guys were studying. And so the first day he was there, some old timer who sort of looked with him down his nose, since he was so young, suggested that he could teach a course. And Pääbo said, I don't think I can do that because I know nothing about the topic. At which point I gave up on bothering him. And so he was a free agent for the rest of his time in Munich. And he spent 8 years there. And he was working already on sequencing so-called ancient DNA. And I'm going to come back to what that really means in a little bit. And then in 1997, the Max Planck Institute, the Max Planck Society, which is sort of like the Hughes Institute of Medicine, very wealthy, and they set up outstanding investigators with their own individual institutes and lots of positions for them to hire assistants into, and a very nice budget to work with. And the only thing was that this being in 97, 98, This was shortly after the wall had come down, and so the Max Planck was interested in having one of its institute in the former East Germany. So after back and forth, they finally decided that Leipzig was a good location, and that's where he did all the work for which he was awarded the Nobel Prize. Most recently, a couple of years ago, he moved to Okinawa, to the Institute of Science and Technology. I know nothing about that move or why he went there, but anyway, that's apparently where he is at the present time. So, this whole talk is about sequencing DNA. And I thought I would start at ground level zero, not assume any particular technical knowledge, and just work our way up into the process. And so we all know that DNA carries the genetic information, and it's a very simple molecule. There's only 4 components, A, T, G, and C. And the thing that's interesting is that the A is always equal to the amount of T, and the G is always equal to C. And that's because A can pair with T, forming hydrogen bonds, 2 hydrogen bonds, which stabilize this so-called base pair, whereas G and C can form 3 hydrogen bonds in the right configuration. And so it's those hydrogen bonds that hold the double-stranded DNA molecule together. And what we really have are 2 single strands of DNA where the bases are like beads on a string, and they're stacked on top of one another, and they're connected by so-called phosphodiester bridge. Each base is associated with a small sugar molecule, a so-called deoxyribose, and in the 5' position of the deoxyribose, there's a carbon with a phosphorus attached to it, and that phosphorus can then be linked to the next base through the sugar molecule, but the linkage is to the so-called 3' position. So we have 5-3 phosphodiester linkages that holds the single-stranded DNA together, the beads on a string. And the other strand, the so-called complementary strand, is the same with one exception. And that is that, of course, everywhere there's a G on the first strand, there's a C and a T and an A, etc. But the important thing is that the complementary strand is in an anti-parallel orientation to the first strand. And that means that the last component here has a 3' hydroxyl group, and the 5' phosphate is down here, whereas this one goes from the 5' phosphate to the 3, etc. So, anti-parallel, that's the key word. And then they form a double helix, and the genius of Watson and Crick, the breakthrough, was when they figured out that in this double helical structure, that the base pairs must be on the inside of the helix, and the phosphodiester bridges on the outside, and that's good because the phosphodiester bridges are highly charged and therefore interact well with the water surrounding the molecule. So here's the conundrum. We got 4 components. In the human genome, there's 1 billion or so or more base pairs. And the challenge is, how do you know which base pair is where? And the reason this is obviously important is that the DNA contains the code. The code actually encodes for amino acids, and it takes 3 bases to code for a single amino acid. And the question is, how do you determine the sequence of all billion or so base pairs when you got so little to start with? And that has been one of the great advances in technology. When I started back in the very late 50s, early 60s, if we could sequence 8 base pairs in a piece of DNA, we were doing very well. And now we can sequence over 1 billion. So that implies a fair amount of progress in the field. And I thought I'd show you a picture of some of the people who were involved in the discovery of important steps. This is Fred Sanger, who invented the basic technique for DNA sequencing. I must say that Wally Gilbert at Harvard came up with a similar method, but turns out that Fred's was much easier to use and scalable. So it's Fred's technique that's used. Now the problem is, as you'll see, when you sequence a piece of DNA, you get out a pattern which if you can read it looking at, let's say, a film out for 100 bases, you're doing well. So we're a long way from 1 billion bases. And the next person here is Lee Hood. A certain amount of sympathy for Lee because he was a classmate at medical school and still a good friend. But Lee realized that in order to make sequencing work, so you could sequence really long pieces of DNA, it had to be automated. And so he invented a machine that sequenced DNA automatically. And that was a really major breakthrough. Now, the other people here come sort of at the end of the story. Sequencing got really pretty good. And so a group of scientists decided that since we were doing so well in sequencing, we ought to really reach out for the stars, as it were, and sequence the whole human genome, because after all, that was the end point for our biological studies. You want to know about people. And so that really turned out to be a major project on a scale with the atomic bomb project during World War II. And it turned out there were 2 groups who competed to do this. The first group was at the NIH. It was headed by Francis Collins. You may know Francis's name. He just stepped down as the director of the National Institutes of Health. At that time, he was the head of the Genome Institute. And Francis picked out a large group of his friends, if you were, and gave them a lot of money to buy sequencing machines and to start sequencing long pieces of DNA. And I just want to show one of these people here. This is Eric Lander. Eric Lander, I put here, he's the villain in this story. Eric Lander is the guy that tried to steal the credit from Jennifer Doudna about discovery of CRISPR. He is the director of the Broad Institute, which is a joint institute between Harvard and MIT. And he's not a nice guy. That's all I'll say. And this was shown because it was demonstrated because when Biden became president, he thought he'd really like to have a high-level science advisor. And so what better than to pick somebody from Boston? And so he picked Eric. So Eric became the presidential science advisor and the head of the president's advisory committee on science and technology. And he was a member of the cabinet. And that lasted for about 3 or 4 months until all the people that worked for him complained bitterly that he created an environment that was impossible to work in. The story that we've heard in many cases in recent years. And it turned out that Eric thought that he was the same as Governor Cuomo in New York and could say anything to anybody. And that didn't last well in the Biden administration. So Eric was swiftly gone and back in Boston, where he still is. You would have what? I think this is a good picture of Eric. It's perfect. Looks like a young Einstein in some ways, if you think about it. Now, the other two people up here are Craig Venter and Hamilton Smith. And Craig Venter had been a scientist at the NIH. He was interested in sequencing human DNA, and he sequenced a lot of short sequences, but he wanted to patent them. And the NIH didn't want to patent them, and so there was a disagreement between Craig and the NIH, and Craig went off and started working with private money, which is an interesting story in itself. And so he was determined he was going to sequence the human genome without all the help from the NIH. But the trouble was he wasn't really quite sure how to do that. And one night at a scientific meeting, he ran into this fellow, Ham Smith, in the bar, and they had a nice conversation. And Smith told him how he could sequence the human genome, and they did. So that was really a kind of a nice story in many ways. But there was very fierce competition between the privately funded group and the public group. And it continued hot and heavy, and it was very public. But finally, in 1990, both of them essentially had the human genome sequence. So President Clinton had a big press conference at the White House. Both groups were invited, and it was announced that American scientists had sequenced the human genome. And of course, some scientists from other countries had contributed as well. Now, what you want to think about is sequencing the human genome is a major step forward. No Nobel Prize has ever been given for this. And I think that reflects the bitter competition between the two groups, and the Nobel Prize Committee just doesn't want to get into the middle of it, so they have bypassed it. Okay. Another Nobel Prize and a major step forward was done by Kary Mullis. Kary was a scientist at a private company, and he invented, when he had a vision one evening, so-called PCR. Now, we've all heard about PCR because that's the technique that's used to see whether or not we have COVID, right? But that's in a slightly different version than what I'm showing you here. And what Kary realized was that if you took a DNA double helix and you heated it up to about 95 degrees centigrade, just below boiling, you would break the hydrogen bonds and you would separate the two complementary strands. So you're left with two single strands. And then you want to add primer. What's primer? Primer is a short stretch of DNA, which is complementary to one of the ends of these single strands. It can base pair and form a stable structure at 55 degrees centigrade. And then DNA polymerase can add the individual bases and synthesize DNA. But the thing about DNA polymerase is it doesn't work if there isn't a primer. So what does this mean? Well, it's very interesting. You had one double helix and you copy each of the individual strands and you wind up with two double helices or 4 DNA strands. Now, if you repeat this 20 or 30 times, you get to a huge number of DNA molecules. And that's the power of PCR. Okay. So here's the DNA replication system in a nutshell, more or less. And that is that you take a collection of DNA molecules, relatively short fragments, and you make a lot of them and you sequence them. And each fragment you determine the sequence of, and the way the system is set up is that you start sequencing overlapping sequences. Not every fragment is exactly in register with the others. And you build up a structure, sort of like a Lego set, and where you might have 50 base pairs in an original fragment, you can wind up with 1 billion if you do it correctly. This is a slight simplification, but good enough for our purposes here. And now we come to Fred Sanger and what he did. What Fred realized was that in the DNA synthesis reaction, if he added, in addition to, let's say, T, he also added something called dideoxy-T. Now, dideoxy-T is what we call a chain ender. DNA polymerase can insert it, but it can't elongate more DNA because the dideoxy is missing a critical component. And so you rig the ratio such that you synthesize a lot of fragments. They end at random T's because every so often a T will be substituted and that'll be the end of the synthesis. And so you get chain termination. And at the end, this dideoxy that they put in carries a fluorescent marker so you can detect it under the right conditions. And you do this with all four of the bases. And you run out all these products on the equivalent of a gel. And you separate all the fragments by how long they are. And then you can read the individual fragments as to whether they were a T or an A or a G or a C. And it comes out looking like this. Here we go. All right. So this is what it really looks like. Here's the T. And so here at the 2 position, there was a T. Every now and then the chain stopped at that one. But sometimes it went further and didn't stop until the 4th T. are up here. The fourth, the second, the third, the fourth, the sixth. And you can do that for each of the individual phases. And you wind up with this stepladder like pattern. And all of us can read the sequence here, right? A, T, A, C, T, C, A, a G. Okay. So that's all there is to DNA sequencing, except that's a little more sophisticated technically. Yeah, I want to go back. Okay. So all of this sequencing gets done in a machine that looks like this, not very big. It's really quite impressive. And in fact, they have small sequencers, which are the size of your iPhone. I mean, this technology is really sophisticated. Okay. So now we can sequence the human genome. The problem is we've already sequenced. By the way, let's talk about the human genome one step further. You know, everybody's DNA is slightly different. Otherwise, we'd all look the same. So, an interesting question is whose DNA was actually sequenced? And that's always been one of the unsolved mysteries, although we all think we know the answers. And the thought is that the DNA that the NIH sequenced was that of Jim Watson, because he had been in charge of this project originally at the DNA, at the NIH. And of course, in the case of Craig Venter, everybody was sure that Craig had sequenced his own DNA. What could be more interesting, right? So those are the two sequences. And now, of course, you can go to 23andMe, and they'll sequence a lot of your DNA for just a few dollars. It's really quite impressive when you think about it. Now, they're not sequencing your whole genome, but they're sequencing a lot of your genome. And from what I understand, they're pretty accurate in terms of the actual sequencing. Now, what the implications of the sequence they get is another question. It's not totally always certain. So let's talk about human DNA. It turns out there's DNA in two places in the human cell, right? One is obviously in the nucleus, in the chromosomes, but the other one turns out to be in the mitochondria. Now, the mitochondria are extra nuclear organelles in the cytoplasm of the cell. They look like little bacteria. And the thought is that originally they were bacteria which infected mammalian cells or eukaryotic cells and proved to be so useful that a symbiotic relationship was formed, and they are maintained as a consequence. And what the mitochondria do is they provide the compounds, the so-called high energy compounds like ATP, which allow important biochemical reactions to actually be carried out by the cell. But the interesting thing about the mitochondria is that they have their own DNA genome, and it's small. It's only 16,500 nucleotide base pairs in this double-stranded circular molecule. Now, I'll tell you a little pearl to take home with you and make you think about deep thoughts. That is that all your mitochondrial DNA comes from your mother, because it turns out that the egg has mitochondria, but the sperm don't. And so when you form an embryo, all the mitochondria are maternal. That means that doing an analysis of the evolution of your mitochondrial genome is much, much easier. And what do we find out? Well, not to offend anybody, I don't think, but it turns out that all of us are derived from 5 women who came out of Africa about 50,000 years ago. So, brothers and sisters, hello, you have more relatives than you really appreciated. And this is the direct genetic evidence of that. So now the challenge for Pablo was to actually get a hold of Neanderthal DNA. That's not the easiest thing in the world. It's rather, precious. And actually, it's not DNA per se, but his bones from Neanderthals that various museums have. And it turns out that it's kind of miraculous that over 50 or 100,000 years, the DNA in these bones is reasonably intact. You wouldn't think that would happen because we all know that when somebody gets buried, decay begins and everything gets chewed up. And DNA is an organic compound, and all of the bugs and worms and what have you, fungi, like to eat DNA. So the fact that there's some that's intact is rather fortunate. And of course, that means that those bones were somehow in an environment which was dry and cool. And so the decay process didn't really get going. Now, the other challenge that Pablo had is that PCR is not very specific in some ways. Doesn't care which DNA it's sequencing as long as the primer fits and you can carry out the reaction. And so the biggest threat to carrying out this kind of research is that you'll have contamination. And that contamination usually comes from the investigators themselves, if they touch something or they breathe on the DNA. And so you really have to do the sequencing and the handling under so-called germ-free conditions, done in a clean room. People have to wear special outfits when they work in there, etc. And of course, the real control when they do this stuff is that they run the sequencing reaction with and without the DNA sample they think they put in. And if they get out a perfect good sequence, guess without the DNA they're supposed to put in, it means they had contamination. And so that's the kind of process that they had to go through. Pablo is very meticulous about this. And so, in fact, he was quite successful. And so he sequenced the human, the Neanderthal genome. And the bottom line is we're not too different. Just really maybe 1 or 2 to 3% at the very most. And that's good because we're only about 5% different than a chimpanzee. So we're really very close when they're talking about anthropoid species. And but what was more interesting is that one of the questions was did Neanderthals and current humans ever coexist? And the answer clearly is yes, for all sorts of reasons that archaeologists have found. But even more interesting is that when you start sequencing the genomes, it turns out there are certain sequences, for instance, that are characteristic of a Neanderthal genome, but not normally found in a human genome, current modern human genome. But in fact, some of those sequences are in fact found in various versions of the human genome. And so it turns out that all of us carry around 3 to 4% of sequences which appear to be derived from Neanderthals, which means there was plenty of interaction between the two groups. And I had a professor in anatomy and medical school, who's a very distinguished guy, and he said, if you think that Neanderthals all got wiped out, he said, just ride the subway in New York. He said, you'll find plenty of people that fit the image that we have of what a Neanderthal is supposed to look like. And that's probably true. So the nice thing is that historically, evolution has been done by studying bones. For instance, the first really early human skeleton was found in Ethiopia, so-called Lucy, Australopithecus afarensis, whatever that means. And that was 1 million years old. So you can study evolution by studying the bones. The nice thing is that when you start being able to do it at the DNA level, you're really getting down to the fundamental components, and you can study individual aspects of it. And so here is a tree. Okay, here we are, Homo sapiens. Neanderthals are up here. There's Homo erectus. And then you come down to Australopithecus africanus. And here's afarensis way down here. Okay. Now, the issue is that not only did Pablo discover the sequence for Neanderthals, but it turned out he discovered a whole new species called the Denisov man, which was about as different from us as the Neanderthals are. And they seem to be mostly the bones with their sequences were in northeastern Russia, up in Siberia, under the tundra. And now, recently, there have been stories that even more specimens have been uncovered, which look very closely related to people. So there were lots of efforts in the evolution of people. And we represent possibly the most successful of those efforts. And that tree, as I said, is shown here. Here's a rumor. Notice that Cro-Magnon is a lot bigger than modern man. Neanderthals a little shorter. Here are the skulls. You can't, I might not be able to tell the difference, and I doubt that any of us could. So that's essentially where we are. So in summary, if you really like skeletons, this is a little closer in. Okay. In summary, to repeat, Svante Pääbo received the 2022 Nobel Prize in Medicine and Physiology for sequencing the Neanderthal genome. Now, I want you to look at the skulls that he's holding and ask yourself if you see apparent relationships between Pablo and the specimen. And I think that makes the point that we're all a lot closer together than we might ordinarily think. And so on that high note, I will end and be happy to take any comments or questions that anybody has. Yes, Jim.
Well, let me come out with the
Yeah,
microphone.
help him out.
If you looked at, take a strand of the DNA, I think, it's reasonably long. Is how physically long is it get to the point that if you strung it all out, that we could detect that visually, or is it way, way smaller than
Well,
that?
let's put it this way. We can see it, but we have to use an electron microscope to do that, which is magnifying it by about, let's say, 50,000 fold. And sometimes a little bit of tricks by fattening up a little bit. But people have been looking at DNA in the electron microscope for a long time. The problem was that you can't see it in enough detail to determine the sequence. We had colleagues back in the early days who actually tried to do that using electron microscopy. And it just wasn't good enough to allow that to happen.
Well, and then we're looking at it now and know what the chemistry of each part of it is. But I guess at this point, we're also looking at what does each part of that do in terms of creating an individual.
Okay. The main components are the four bases, A, G, T, and C. That's the genetic code. So leucine, and the amino acid leucine, which is a component part of proteins, is encoded by 3 T's in a row. And the question is, I hate to get into this too deeply, but Francis Crick, who was an authentic genius, invented the so-called dogma of molecular biology essentially off the top of his head. Once he had solved with Watson the structure of DNA, he could see what the implications were. And the question is whether the so-called codons that encode individual amino acids in the protein should be a triplet codon or a doublet would be enough, two bases. And it turns out at one level it has to be 3, because with 2 to the 4th you don't get enough amino acids. I guess it's 4 squared is what I'm really trying to say. Whereas 4 cubed gives you more than enough. So it turns out that sometimes the 3rd position in the triplet can vary and you still get the same amino acid. We don't want to go too far along that road because it leads to all sorts of complications.
Let's go to John on Zoom, and then we'll come back to the Oak Room.
Yeah. I was very relieved that when I gave a Christmas present to my twin brother, that it came back 23andMe that said, "You have a twin," which I knew. But when I came to the University of Florida back in the late 60s, we had a guy here who could take fingerprints which even in twins are not identical, but are so much more alike to one another than any other. And then he just looked at us and said, yeah, you're identical twins. But 23andMe gave me that. I have a question about, did he sequence any Homo sapiens, I mean, modern or Cro-Magnon genes from the same period as the Neanderthal genes? I mean, so was that the comparison?
As far as I know, the answer is yes, but that's not what his book is about. So
Yeah, I got to read the book.
it's hard to
Also,
know.
also, it's my understanding that we have Denisovan or Denisovan, that lineage is a result of a few bones out of a hand, a pinky, of some hand bones, and with that we haven't really found a distinctive skull of that period. So do you think that picture was kind of speculative, or do we have a have we found a skull?
Whether you think DNA lies or not.
Yeah, I don't know. So it's just interesting.
For us true believers, of course.
And then finally, if they sequenced Jim Watson's whole genome, did they find a gene that would explain his somewhat bigoted view of things?
They didn't find the bigoted.
No, okay. I just wondered.
I think the thing about Watson, which we're looking for, is the gene which inhibits you from saying certain things at various times is defective in Watson. And so
Yes,
he sorts out whatever comes across his mind at that moment.
but there are mutations in modern man. Yeah, that probably explains it. Thanks.
Okay, back to the Oak Room here.
Related to Jim's question, since this is all so difficult and small, how does that machine work that can analyze the sequence?
That's what I don't want to get into with you, because I don't have the slightest idea. It's sort of it's called capillary drip analysis. I mean, it's like asking how a transistor works, which gets smaller and smaller and smaller. It's the same kind of basis here. And beyond that, I have no idea. Yeah, Larry.
If our mitochondrial DNA can be traced back to 5 women who got lost coming out of Africa in search of a mall, if you did their DNA mitochondrial, might it go back to one woman who you could designate as Eve?
Well, what you're really asking is, do we all go back to Abraham?
Eve.
Or the equivalent? And I think that's a perfectly valid question to ask. And it might actually be the case, but we don't, we just don't have any knowledge of that. Now, there was an interesting story about a tribe in southern Africa that that looked like they were practicing Judaism. I mean, you looked at their rituals. And they had oral history, which said they had come by the sea from up north, the Arabian Peninsula. And so the question was, were they a lost tribe of Israel? Everybody has sort of laughed at that notion, poo-pooed it, till some smart guy took a look actually at their Y chromosomes and found out that they were very closely related to the Levites of the ancient Israelites. So you don't want to be too skeptical over some of these possibilities when you hear them, as far-fetched as they might seem to you.
Well, why should it stop at 5 when it's coming funnel?
No, I agree completely, but I don't know the answer to your question.
Paul on Zoom, I think it looks like you're unmuted. Did you have a question, Paul?
Well, I'm not sure it's a very good question, but I'll try. when we do this kind of genealogical research using the DNA, as has been quite a rage lately, does any of the DNA evidence or results show differentials within Homo sapien, for instance, or in terms of the brain and how the brain works and how the whole organism works and what are the relationships and are they different depending on whether you talk about Neanderthal or within a species?
You realize that that's a dynamite question.
Oh, I'm glad I found it.
Obvious. But what I'd say is that at the individual level, there's all sorts of genes which are involved in behavior, whether act
Yeah.
to a given racial group or something like that, I think is totally dubious. But that's the concern that people have when you start raising those kinds of questions. Remember, there was a story about trying to identify genes that might be associated with deviant behavior or criminal behavior, being able to see whether some individuals had those genes, especially people who were known criminals. That turned out not to be, they couldn't do that study. I mean, it just raised too many hackles, sociologically.
Any other questions? Well, we thank you again, Ken. John or Ken, do you want to give us a preview of next week?
Next week,
The schedule.
Dan Courage is next.
Courage. Oh, yes. I spoke to John Axe. He's going to be after that, and he's ready and willing. So I'll try to be there in person.
So it turns out that we couldn't get a physicist historically who could give us a talk that anybody could understand. The one physicist that we have is John, who gave a nice course a couple summers ago on astrophysics. And so I convinced John, with some arm twisting, that he could handle the physics prize this year, because the physics prize was described as being given for complex problems. Not even the Nobel Committee was willing to spell out what they really thought it was for. So I saw, I figured if anybody could do this and get us near, it would be John, and he tells me he's been working hard on that.
Well, we'll see how we do with chemistry next week and then physics after that. So thank you again, Ken.
Yes. I really enjoyed that.